let me start again then for the sake of the recording. I'm grateful to you all for joining. I know it's impossible to have a time which is convenient for everybody to be online simultaneously, but fortunately the Zoom has this option of recording, so we will have a recorded video and soundtrack and slides and everything which anybody can look at any time they want into the future. The history of our centre. Why is history important? I think most people agree that history is important, but it's equally true that for political history, it's generally written by the victors or by the survivors. So it doesn't mean it's always very objective. However, in science and academic fields, history can be largely more objective, never completely because it's a human activity, but it can be more objective because in science and medicine, things are published and dated, and there's an objective record of who did what, when, and so on. So that's quite helpful. And I think the history of academic centers and so on is important because it gives you the place where you are, how you arrived in the organization in which you operate, where it came from, where it might be going to, and so on. So I think this is quite an important topic. In this present case, the history of our centers starts indirectly with Dame Sheila Sherlock, Professor Dame Sheila Sherlock, who was the leading liver physician of the 20th century. She was the professor of medicine at the Royal Free Hospital School of Medicine for many years and was undoubtedly the most distinguished person ever to have been on the staff at the Royal Free. She retired, as she was forced to do in those days at the age of 65, and was succeeded by one of her lieutenants. But over a number of years thereafter, as generally happens when there's somebody very innovative and exciting who sets up a world-leading center, things tend to go off the boil a bit, and it's a hard act to follow thereafter. And that's what happened with the Centre for Hepatology at the Royal Free. Now, when the successor to Sheila Sherlock retired, Royal Free, which had then lately been taken over by UCL and merged into UCL's medical school, they struggled to find a hepatologist to succeed her. So let me now share my screen. So here's the question. Why do these centers exist and why are they at the Royal Free? And there's a simple short answer to that, and it is because of me. So in 1998, I had been invited by Cambridge University, my alma mater, to consider the post of head of pathology at Cambridge University, a job which I would have liked to have had since I was aged about 18. But when they offered it to me when I was 55 years old, it was no longer the job that I wanted to do at that time, so I turned it down. Shortly afterwards, UCL invited me to come to be the head of medicine at the Royal Free Campus and establish my own academic and center there in UCL medicine. And I've written my name in full and written FRS afterwards for reasons which I will make clear to you as we go on a bit further. And the invitation they extended was absolutely extraordinary. They were going to build 2 new floors comprising 3,000 square meters at a cost of £9 million, the present lower third and upper third floors on top of the medical school building. And I was asked to design all the space and to allocate it. Exclusively done by me. They gave me a number of senior and less senior academic posts vacant to appoint to, lots of support in various different ways, an option to bring anybody I wanted with me when I joined the institution. So who came? The answer was my whole team, which was then called the Immunological Medicine Unit at the Royal Postgraduate Medical School at Hammersmith, plus 2 other people, Peter Whitehead and Andrea Francis. Peter Whitehead, the nicest, most helpful IT guy you could possibly imagine, was head of computing and so on at the Hammersmith. And the Royal Free had very primitive, if any, infrastructure for computing and digital work at that time. So I recruited Peter to become head of IT at the Royal Free before we moved so that things could be in place by the time we arrived. And Andrea Francis is a wonderfully talented physiotherapist. And I needed her for personal reasons, which I'll illustrate here. I used to be a very sporty guy, and 2 of my favorite activities were surfing in Maui and powder skiing in Utah. And I was often injured as a result of this, as you can see. Well, you won't actually see it, but I'll show you on the next slide something more about it. But I often needed Andrea to keep me together and put me back into one piece. So here is skiing in the powder in Wyoming. Unfortunately, in this little clip, which I'm sharing with you, no injuries were sustained. Nobody was hurt in the making of this film. Okay, so why do these centers exist, the CAAPP and the NAC? Well, in 1999, I named the center, the overarching center, for my scientific and clinical work, which I've been doing since 1975. That's where the name came from. So in the early 70s, I had worked on the key properties of SAP and CRP, the so-called pentraxin protein family, which I identified at that time, and crucially discovered the calcium-dependent binding of SAP to amyloid fibrils. In the early 80s, we elucidated the reasons for the universal presence of SAP in amyloid deposits. And that led me to my invention of SAP scintigraphy in the mid-80s. And over the next few years, we developed that into a clinical tool. And this had an transformative impact on knowledge and practice in amyloidosis, which led to the center becoming the de facto national referral center for amyloidosis over the following decade. So I'm going to just show you a few of the papers which were seminal in all this story. This is the first paper showing the calcium-dependent binding property of SAP. Okay, and I want to also point out that I also often added a last sentence to the summaries of many of our papers, which presaged what we thought the new observations we were reporting could lead to. So in this case, we said the presence of P component in all forms of amyloid may be due to its calcium-dependent binding. Okay, and so the next thing that came after that was indeed the calcium-dependent binding of SAP to amyloid fibrils, which you hadn't discovered when the original paper was written. And here it is demonstrated there. And although we didn't know what the normal function of SAP was, this binding suggested that it could have a role in the deposition or persistence of amyloid. Okay, and then we showed the presence of this pentraxin family throughout vertebrate phylogeny and beyond, and different biological behaviors of pentraxin proteins in different species. The first paper showing a possible association between the amount of SAP that animals produced and whether they got amyloidosis or not. The discovery that SAP was a normal tissue protein as well as a blood protein in the elastic fibers here and previously in the glomerular basement membrane. And this led to the invention of SAP scintigraphy. So in the early 80s, it was obvious to me that just doing tissue biopsies, which is the way amyloid only way the amyloid could be diagnosed in those days, it's a tiny sample. It doesn't tell you anything that you need other than that the patient's got amyloidosis and that you might be able to find out what type it is. But it doesn't tell you where the amyloid is, whether it's getting more or less, whether it's getting more or less with treatment or not. So we were in the dark, we were blind. And our metabolic studies of SAP in the mouse, demonstrating that circulating SAP is the precursor of SAP in amyloid deposits, pointed the way very clearly to a different approach. And that led me in 1985-86 to the first demonstration that radio-labeled SAP localized specifically and can be imaged in amyloid deposits in experimental mice in vivo. So here's the original metabolic studies, which showed the precursor product relationship of SAP and amyloid. And if you look at the last sentence of the abstract, the present results suggest a means for selective targeting of diagnostic traces and/or effector agents to amyloid deposits in vivo, predictions which we abundantly fulfilled over the next decades. And here's the first paper reporting my invention of SAP scintigraphy. Okay, in the final sentence, these findings suggest that such imaging may have applications for the diagnosis and quantitation of amyloid deposits in humans. This paper was submitted in October 1986. And Dan Caspi, who was the brightest medical doctoral fellow that I've ever had, played a seminal role in the work leading up to and to this paper itself. Now, there's a nice story around this. Having done this work, I needed somebody able, intelligent, industrious to work full-time, flat out on transferring the imaging from mice to patients. And there was a very bright registrar at Hammersmith then called John Saville, And I invited him to come and be my PhD student to do this project. Sadly, he turned me down because he had already accepted a PhD offer from Chris Hazlitt, another consultant at the Hammersmith, and so he wasn't able to accept the invitation. But unsurprisingly, John Saville went on to great things. He became the principal of the Edinburgh Medical School. He became the chief executive of the Medical Research Council. He was knighted and became a fellow of the Royal Society. And a few years ago, we met at a meeting at the Academy of Medical Sciences where we had a conversation, which he then confirmed to me in this nice letter that he wrote afterwards. He wrote to confirm that to the best of his knowledge, I was the current record holder for continuous support from the Medical Research Council, 47 years, from 1969 to 2016. And then with a typical sense of humor, he says, I suspect this unblemished record has something to do with not having taken me on as a PhD student in 1986. And instead, I took on Philip, and he started his PhD studies with me at that time. And I very closely directed and supervised all the work developing and implementing labeled SAP studies in patients and other studies related to that. And curiously and interestingly, I personally have either isolated or directed the isolation of all the human SAP that has ever been used by our group, including the material currently in routine use for SAP scintigraphy, all done using methods that I invented, developed, and perfected. And crucially for the NAC, the NHS has reimbursed this unique contribution of expertise, time, and effort that I put into making the current batch of tracer SAP sufficiently to provide the SAP with very substantial funding. Now, the first publication we had of human SAP scintigraphy was in the Lancet, shown here. And the first few patients that we illustrated here had clinical amyloid hepatomegaly. And you can see that as soon as you inject the tracer into them, it rapidly disappears from the blood because it's sticking to the massive amyloid in the liver. Now, the very first patient who ever had SAP scintigraphy done was a patient with massive amyloid hepatomegaly. We put him on the scintigraphy table, the scanner table. Philip injected the radioactive tracer into him, and the images were registered in real time. Instantly, the liver became red hot. Philip said, "Oh dear, it's failed. The stuff must be aggregated or denatured." In contrast, I said, "Yes, it's really worked. It's doing exactly what it should do." Of course, I had the advantage at that stage of a lot of experience of radio-labeled tracer studies in animals, and I knew a lot about the avid specific binding of SAP to amyloid, and I knew that radio-labeled amyloid, SAP rather, would stick specifically and stay in the amyloid deposits, and that's exactly what it did. Over the next few years, we worked hard characterizing SAP scintigraphy in many different situations and ended up with these two landmark papers which describe the application clinically. Scintigraphy after the injection of labeled SAP can be used for diagnosing, locating, and monitoring the extent of systemic amyloidosis. And that's what it's turned out to be, 50 or 60,000 scans later, none of which have had any single adverse effect in any patient. So this had a transformative impact, funded entirely by my MRC program grant research funding. From 1979 to 1999, 20 years, we attracted steadily increasing number of patient referrals to the immunological medicine unit because of the availability of SAP scintigraphy. And after Philip had finished his PhD, I kept him attached to the unit while he continued his clinical training towards a very early consultant appointment and other promotions, which I enthusiastically did for him. And I progressively delegated the care of the amyloid patients to him because I wanted to concentrate on my research, mostly laboratory-based, some of it clinical, and I delegated to him the patient care. And the IMU thus became the de facto national referral center for amyloidosis. Of course, a lot of other work went on, clinical and scientific, in the IMU in those years. We pioneered the clinical application of CRP measurement including in cardiovascular disease, which I'll say a little bit more about in a moment. We made some international reference standards at the invitation of the WHO for CRP, serum amyloid P component, and thyroxine binding globulin. We made seminal observations of amyloid fibril precursor product relationships. We identified multiple mutations in different amyloidogenic proteins that cause hereditary amyloidosis. We elucidated the structure, the biology, and the pathobiology of SAP. We validated SAP and CRP as therapeutic targets. I patented the invention of a high throughput screen for small molecule SAP ligands and went on to invent a drug targeting SAP together with Roche. I'll show you just a couple of examples of some of this work. Here is a seminal key original observation relating SAA production in inflammatory diseases to the risk of reactive systemic AA amyloidosis. And here is the predictive last sentence again. Prospective monitoring of SAA concentration in predisposing diseases may help to identify those individuals who are most at risk for amyloidosis. Few people, if any, were thinking along those lines at that time. This was a seminal observation which turned out to be true, and not just for AA amyloidosis, but for all types of amyloidosis. The lysozyme story, on which I will dwell very briefly, was absolutely critical in understanding of amyloid fibrillogenesis. In the early 80s, a couple of families were referred to me who had hereditary systemic amyloidosis. It was autosomal dominant. It was obvious that they had a mutation in a gene for a protein which must be making it amyloidogenic. That was the obvious implication. The very last one of these surviving patients, everybody else had died out, was coming to the end of his life, and we wanted to have an autopsy when the patient died. He was in Birmingham, and the clinicians looking after him agreed that they would undertake an autopsy. And I said, but crucially, we must have the organs, in whole organs, not just a biopsy or a piece, but the whole organs, in order to be able to extract enough amyloid fibrils from them to be able to identify the protein that is causing the amyloidosis. And they said, yes, yes, we understand. I said, ring me as soon as the autopsy is underway. If necessary, I will come personally and pick up the organs, or else we'll find the right way to get them safely transferred to the Hammersmith. They said, yes, yes, we'll do that. The patient died. They did the autopsy. They rang me up and they said, it's all done. The autopsy's done. The tissue samples are on their way to you. I said, what? The whole heart, liver, spleen, kidneys full of amyloid? How are you sending them? In the post, they said. You must be joking. No, we've sent them. I said, how much have you sent? Oh, they're about, 1 cubic centimeter of each organ. I said, I told you explicitly I wanted the whole organs. So these tiny little fragments, the size of my fingertip, arrived at the Hammersmith the next day. And all we could do with them was put them in the freezer, because in order to sequence a protein in that era, you had to have about 30 milligrams of purified single protein. There was no way we were going to get that from these tiny little fragments of tissue. So nothing happened for a number of years. Fast forward 10 years or so, and the technology advanced. You could then do an N-terminal sequence on a protein from a band in a gel containing 20 or 30 micrograms of protein, never mind milligrams. So I invented a new technique for isolating amyloid fibrils from these tiny amounts of amyloidotic tissue. And we got a tiny amount of fibrils, we ran the gel, we sequenced the protein, and it was lysozyme. And when I had the phone call telling me that in my office, I danced into the lab, waving my arms in the air and said, it's lysozyme, it's lysozyme. And all my team in the lab, Philip and the others, they all looked at me and said, what are you so excited about? What's lysozyme? So now I'm going to ask everybody here a question. Those of you who already know this story can stay silent. I want anybody who hasn't heard this story before, who wants to speak up and unmute themselves, tell me why I was so excited that it was lysozyme. Let's see if anybody wants to respond. It doesn't sound as if anybody wants to speak up. So let me tell you. Lysozyme was about the third protein ever to have its crystal structure solved. It's been used as a model protein for all sorts of protein studies, particularly protein folding and misfolding, but also lots of immunology and all sorts of things. It's the ideal model protein. It was certain that if we discovered something significant that made the lysozyme amyloidogenic in these patients, we would learn something fundamental about amyloidosis. But there's another reason why it's fantastically important, and this is a historical reason. Does anybody know who discovered lysozyme? No responders? Well, the answer is it was Sir Alexander Fleming. He discovered lysozyme many years before he discovered penicillin. He was culturing bacteria from his nose, and he had got a culture plate with bacteria sitting on it, bacterial colonies, and he had a streaming cold. And some snot from his nose dropped onto the plate. And where it dropped onto one of the bacterial colonies, the colony of bacteria dissolved. And he investigated this as a proper scientist. What is it in nasal secretions that dissolve this particular bacterium? And he found it was an enzyme. And this enzyme was ubiquitous. It's in tears. It's in saliva. It's in most living creatures produce something that does this action of lysozyme, okay? And nobody to this day knows what the real function of lysozyme is and so on, but it's there and it's ubiquitous. But he had discovered for the first time, a biological product that killed bacteria and dissolved bacteria. And when many later he saw the penicillium mold growing next to Staph aureus colonies and producing something that dissolved the bacteria, his mind was prepared for the sensational discovery of penicillin. So lysozyme is critical in one of the most important biomedical discoveries of all time, the discovery of penicillin. And here we have it as an amyloid fibril protein. And why is it an amyloid fibril protein? Because it has mutations in it, a couple of single point mutations, single residue substitutions in the two different families, which encode amyloidogenicity. And our final statement here is our observations should provide a powerful model for understanding amyloidogenesis. And a few years later, this seminal paper, which is where Vittorio Bellotti first made his impact within our laboratory. He was already an expert on amyloid, but here he made a seminal impact in the whole world of amyloidosis by showing that these amyloidogenic mutations destabilize lysozyme so that it more easily misfolds and can then aggregate specifically as amyloid fibrils. A really, really important contribution to amyloidosis. This paper has been cited over 1,000 times. So why did they invite me to the Royal Free in 1998? I've given you a history of what went on before we came to the Royal Free. Why did they invite me? Well, I had a track record. In 1970 to 73, I did my PhD in immunology in Cambridge, where without a supervisor's project, I devised my own project and led to a fundamental new discovery in immunology, which led to later partly to my election to the Royal Society. I published 4 Nature papers initially from that thesis, 2 of which as a sole author and about 20 other papers later. I was elected to a fellowship at Trinity College. I'll just tell you a bit about Trinity College because that's also part of the track record. Trinity College, for those of you who don't know, it is the biggest college in Oxford or Cambridge, but it's very small. It's got about 900 students at any one time, undergraduates and something over 100 fellows. From this tiny academic institution, it has produced 34 Nobel Prizes. That is more than any other country in the world except for 4. It's about 1/3 of all British Nobel Prizes for science, and there are only 4 whole countries in the world that have gotten more Nobel Prizes than Trinity. A pretty unique institution. And my track record over the years then and subsequently have been recognized by prestigious named awards from the Royal Colleges of Physicians, pathologists, surgeons. I was a founder fellow of the Academy of Medical Sciences, and I was a fellow of the Royal Society. The Royal Society, for those of you who don't know anything about it, is the National Academy of Science of the UK, founded in 1660. It's the oldest, by far the most distinguished national academy of that sort. Everybody great in British science has been a fellow of the Royal Society. It's like a scientific knighthood. That's the best way to explain it, to be a fellow of the Royal Society. The Royal Free Hospital School of Medicine, apart from Sheila Sherlock, who was outstanding, was not the most distinguished academic medical school in amongst the medical schools in the UK and in London. In the whole history of the Royal Free, it's had 4 Fellows of the Royal Society. Myself, a man called Dennis Chapman, a man called Geoffrey Burnstock, who was a FRS at UCL, and when he retired, came and worked at the Royal Free, and Dame Sheila Sherlock herself. Now, tragically, she was not elected to the Royal Society when she was at the peak of her powers. Lord Todd, the Nobel laureate for chemistry, who was then the president of the Royal Society, when her election came up, he said, well, she's a doctor. She just does what doctors do. She doesn't need to be a fellow of the Royal Society. So she was not elected. A disgraceful bit of misogyny and anti-doctorism. The first thing I did when I was elected a fellow of the Royal Society was to nominate Sheila Sherlock, who was then very old and not very well. And she was elected immediately the first year, which is extremely unusual, but she was elected, thank God, over a year before she sadly passed away. And she was ecstatic about it. It was the thing that she wanted more than anything else, and she was delighted to have it. So I was invited to the Royal Free based on this track record. And what did I bring? Well, I told you I sent ahead Peter White ahead and Andrea Francis. I brought the whole team, the IMU team and our grant-funded equipment, all my long-standing MRC and other substantial research funding. I appointed Philip a chair in medicine to run the clinical amyloidosis service, a chair in medicine for Humphrey Hodgson to run hepatology and the undergraduate teaching. Helen and Julian had meanwhile applied to me for their MD clinical research training, which I delegated to Philip. He supervised that. Julian then applied for a PhD under my supervision. And after completion of their clinical training, both at the Royal Free and elsewhere, we appointed them as consultants in the NAC. So that's what we've brought. The new centre was named, as I said, by me for my research fields. That's the overarching centre. Now, before 1998, the NHS system for tertiary and quaternary referrals was hopelessly inadequate. CMO, the brilliant CMO, Sally Davis, Chief Medical Officer, in 1998, she introduced direct Department of Health funding for highly specialised services. And as soon as that was started, I applied initially from the Hammersmith for such a support for amyloidosis, but it was rejected. In 1999, after the move to Royal Free, I applied again successfully, and we were funded from then and until now. I invented the name National Amyloidosis Centre. I appointed Philip to the post of clinical director and delegated to him running the National Amyloidosis Centre from the clinical standpoint. Thus, the NAC was born. What has the NAC done? Well, this is ground with which you're all much more familiar, but it became the world-leading specialist amyloidosis centre with the largest, most diverse cohort of patients anywhere in the world. In 1999, we were 8 staff, 300 patient visits a year approximately. By 2019, more than 60 staff, more than 5,000 annual patient visits. The unique routine availability of SAP scintigraphy remains our distinctive feature. But also, as you know, we have the full range of amyloidosis-associated investigative and diagnostic expertise and expert management advice. And Philip's leading expertise in autoinflammatory disease has extended into that area too. As you know, these inflammatory diseases are strong risk factors for AA amyloidosis. And all of this is funded directly by the NHS on a per capita basis. The overall achievement, decades of MRC funding to me, from starting in 1969 and eventually leading to the transformative routine SAP scintigraphy clinical test and service, is a triumph of British biomedicine and has been extensively recognized as such. Now, what have I done in the 20 years at the Royal Free specifically related to the NAC? Well, in addition to obviously creating it and supporting it throughout, I've been instrumental in selecting, appointing and training and mentoring the initial clinical staff. Other activities have been the Amyloidosis Research Fund. As soon as the NAC got underway, grateful patients rushed to donate money. They wanted a charity they could donate money to that would exclusively support our work in the NAC. And I set that Amyloidosis Research Fund initially in the UCL system for many years, and then lately in the last few years transferred to the Royal Free Charity. And it was managed and administered amazingly well by Beth Jones, who had been my secretary and PA since 1986, so been up with us for a very long time. I set up in about 2013 the Amyloidosis Awareness Program. All the people in NAC are very familiar with this, all the patient leaflets, the newsletters, the website, which and so on, which are greatly appreciated by patients of ours and patients all over the world. That is run by my daughter, Miriam Vered, who, in addition to having a triple first-class degree in medical sciences tripos from Trinity College, Cambridge, and coming second in the whole of the University of London in her medical finals, in which, by the way, she was excellently taught by Julian, who's a good medical teacher. She also has her own medical consultancy firm, medical communications firm, and she has done an absolutely brilliant professional job in running this much appreciated program. I got Peter Whitehead many years ago to start establishing an amyloidosis patient database, and this has been taken forward by subsequent people in IT, most recently, of course, Islam, but many people have contributed to this. And it's an exceptional resource because of the diversity and size of our patient base. Proteomic mass spectrometry was established in our center by Graham. Graham Taylor, been my colleague, co-grant holder, friend for many years at Hammersmith before we came. When he left the Hammersmith in about 2005, I think, or 2006, first thing he did was come and see me. And the first thing I said was, come and work for me, don't retire. Come and work here. And he immediately did. And he is a great mass spectrometry expert, together with Nigel, his close colleague, and Janet, and latterly Diana. Proteomic mass spectrometry in our center leads the world. Maybe not in number of cases, but certainly in terms of expertise and understanding. PPI, patient and public involvement, crucial for the NHS. I initiated an activity in that, ran it for a number of years, retired from it a while back. I don't know where it's going now, but it's something that ought to be happening. And finally, I've had a couple of really great original ideas based on my own work and also on the work of Vittorio Bellotti, and that is to look at the genomics related to complement and the thrombolytic pathway in patients with amyloidosis. I set up with great effort high-level collaboration with genomic material and genomic expertise, and I handed this program off to Julian, and I would like to have him keep me fully updated in a timely fashion about how that program is going. It has the capacity to contribute enormously to understanding and risk of amyloidosis and eventually its treatment. Okay, so what about the laboratory now, as opposed to the NAC? The CAAPP from 1999 to 2011 comprised my own research laboratories and the NAC under this umbrella. In 2006, the laboratory side was expanded by the recruitment of Paul Simmons and his wife Rhea, who joined the unit. Paul had been a junior lecturer in anatomy, was made redundant by the anatomy department down at UCL. So I rescued him from that and set him up as the genetics unit in our center. And they did an outstandingly good job in collaborating with us on many mouse transgenic projects and so on. In 2007, I recruited to a new laboratory of protein crystallography, 2 professors, Steve Wood and John Cooper, and a senior lecturer, Alan Coker. They had been together at Birkbeck College working with Tom Blundell, with whom in the 1980s, we as a group solved the structure of human SAP by X-ray crystallography, another seminal landmark achievement. And we had continued to collaborate, particularly Steve and I, in the closest possible way. All the structural biology through crystallography that I did was done by Steve and with Steve. And so I set up this laboratory for them to come and work with us at UCL in our center. In 2008, the UCL evacuated the second floor of the medical school, and I negotiated successfully to acquire it for the Division of Medicine. I raised £6.5 million to renovate and expand it, including expanding the lower third NAC and the laboratories, and this opened in 2011. At that point, I was thinking very seriously about changing my own direction. I did not any longer want to be running a large department of medicine at the Royal Free. I wanted to focus on my own research, particularly drug discovery and development, which was going very well. And I recruited Professor Vittorio Bellotti from Pavia in Italy via an MRC strategic award to come and work in our center and to eventually be my successor as head of the labs. And so in 2011, I retired as head of medicine to become director of what I newly called the Wolfson Drug Discovery Unit, to recognize £2 million that the Wolfson Foundation had given to extend and expand our labs. So it was just a rebadging job. Here's the letter that Malcolm Grant, the provost and president of UCL, wrote to John Saville, the chief executive of the MRC, soliciting this strategic award to fund Vittorio's recruitment to our center. And you can see I've highlighted some of the key bits of it, but it just recites again many of the things that I have been telling you about. Here's the recruitment plan. I would retire, Vittorio would succeed me, etc., etc., etc. That was the plan. So what did we do? What have we achieved in the, after that era? And the answer is quite a lot, particularly in the region of therapeutic drugs, identifying targets, and developing drugs. Miridisap and SAP depletion, I'll show you some work on that. The DESPIAD trial of Miridisap, the SAP depleting drug in Alzheimer's disease. The invention of antibody immunotherapy against SAP to remove systemic amyloid deposits in a multi-million-pound licensing deal with GSK. Lots of work on CRP in cardiovascular disease. The patented invention of CRP inhibitor compounds. The first in human clinical trial of a real drug like CRP inhibitor is led by Apollo Therapeutics, and we hope it will be happening soon. We've invented so-called TTR superstabilizer compounds to not only arrest TTR amyloidosis, but also prevent it, acting like statins for this horrible disease. And finally, Vittorio Bellotti has made absolutely seminal discoveries of the critical role of mechanical and mechanoenzymatic factors in amyloid fibrillogenesis of all types. So I'll just illustrate some of this. Here's the paper, which was a medicinal chemistry highlight of 2002, according to the American Chemical Society, our invention of Miridisap, the SAP depleting drug. Here's the drug. CPHPC is what I originally called it, just this acronym to designate its chemical structure. And you can see here the complex of 2 SAP molecules cross-linked by 5 drug molecules. That complex is instantly cleared from the circulation. It's taken up in the liver, and that process removes some SAP from amyloid deposit, but it doesn't remove all the SAP from the amyloid deposits. I always hoped and still hope that if you could remove all the SAP from amyloid, the body would see the amyloid as abnormal and get rid of it. But it didn't. We couldn't test this because our drug did not remove all SAP from amyloid deposits. I'll come back to that in a moment. However, the drug does remove all the SAP from the CSF that bathes the brain and from the small amounts of amyloid which are present in the brain in Alzheimer's disease. And this work with Martin Rosser, the leading UK dementia physician, demonstrated exquisitely well how potent this drug could be. And that led to our most recent work, which I show here, published just a month or 2 ago, where we measured the SAP content in the brain of old people who are dying in a study where we know whether they were demented at death or not. The association between the amount of SAP in the brain of patients dying with dementia is astonishingly significant. If you're in the top tertile of SAP content in your brain, when you die, you have a 5-fold increased risk of being demented at death. Whereas if your SAP is normal or only modestly elevated, you don't have an increased risk. And this is true across all levels of classical neuropathology. It's independent of anything else that's going on in the brain. It's the most significant predictor of whether somebody will be demented when they die. And as we say in our predictive sentence, this finding is consistent with a pathogenetic role in dementia, a pathogenetic role for SAP, for which we have a safe, well-tolerated drug that removes all the SAP from the brain. So if you believe in betting in the horses, on horses with a track record, you might not want to bet against our track record here in relation to these predictive assertions. Who knows? We hope to find out. So SAP removal, the best we could do with miridesap, did not cure amyloidosis, but it led me to another invention. So in 2005, I figured out that we could remove SAP from the blood, leave some SAP in the amyloid, and then safely use anti-SAP antibodies to target the amyloid deposits, to activate complement, to trigger macrophages to clear away the amyloid. So that was the invention, and it works miraculously well. Here's the Nature paper where we reported this in mice. Here's a control mouse. All the spleen is more or less replaced by this amyloid. The green stuff is the amyloid. After one shot of antibody, a few weeks later, virtually no amyloid left. Miraculous. And how does it happen? By macrophages gobbling up the amyloid. So here's the amyloid with no cells taking any interest in it before you do any treatment. One day after antibody, every bit of amyloid surrounded by macrophages, very interested in the amyloid. A few days later, all the macrophages have fused into multinucleated giant cells. All the amyloid is inside them. They've gobbled it up and they are destroying it. 2 weeks later, all gone. No abnormal cells, no amyloid. Here you see in the EM, the same thing, extracellular amyloid, fibrillar amyloid, no cells taking any interest in it at all. Here, after antibody, all the amyloid is inside the cytoplasm of these multinucleated giant cells, many nuclei, huge phagolysosomes, amyloid being digested and degraded. And later we studied the phenotype of multinucleated giant cells in vitro for the first time and showed that they had all the right properties to do exactly what we had triggered them deliberately, designed to do in vivo. And this work in mice predicted exactly what happened in patients. New England Journal paper, therapeutic clearance of amyloid by antibodies to SAP. Here's a patient with massive amyloid hepatomegaly. 42 days after a single dose of miridesap plus anti-SAP antibody, amyloid all gone. And all the relevant parameters that you can measure alongside that support the SAP scintigraphy. It works. Repeat doses in some patients were needed. One dose wasn't enough. Repeat dosing eventually gets rid of most or all of the amyloid. And it's good for the patients. You don't need to look at this. It's just a placeholder to remind me to tell you that the liver function got better when we got rid of the amyloid, unsurprisingly. So sadly, GSK abandoned this, as big pharma companies often want to do. The head of research changed, so Patrick Vallance went off to become the chief government scientific advisor. You'll all be familiar with him. And the chap who replaced him decided to divest GSK of all their rare disease portfolio. They also designed a very badly designed phase 2 clinical trial, which I told them repeatedly was designed to fail. But they ignored that and they went ahead with it. And we only had one patient with cardiac amyloidosis in that trial. It was a cardiac amyloidosis trial. We had one patient where there was objective evidence of some regression of amyloid from the heart, but not in any of the others. I think I know the probable reason why, but we never had the opportunity to chase that out. And the whole program got shut down in a way that disabled us from following it up. So that was the end of that little story. But the proof of concept that the right antibodies can make amyloid go away, we proved that, and more is to come in that pathway in due course. CRP and cardiovascular disease, very important. A huge area of work, very controversial, to which we have made massive contributions. I started it all off in a New England paper published in 1993 with Professor Attilio Mazzari at the Hammersmith. A paper which has been cited 3,500 times and it set off this whole field. I haven't got time to talk more detail about it, but anybody who's interested, here are all our papers on the subject, and the important ones, the really important ones, are in bold if you want to read about it. What else did we do? We identified CRP as a therapeutic target in acute myocardial infarction and probably many other conditions. Here is the first molecule that we designed, invented, a new chemical entity, bisphosphocholene hexane, which cross-links CRP molecules just like miridesat cross-links SAP molecules. And we went on to show that this compound completely blocked the pathogenetic effects of human CRP in a rat model of acute myocardial infarction. You tie the coronary artery in a rat, it gets a heart attack, a certain amount of the heart muscle is infarcted, is dead. Here it is, that's what happens when you do it. If you give human CRP to these rats, it makes the infarct much worse. If you give human CRP but follow it up with our drug, our inhibitor, it completely abrogates the pathogenic effect. And anybody who's come to our center will have seen this image of the cover of Nature, where we reported this in 2006. Now, it's pretty difficult to make a drug. Those molecules that we originally invented could not, for chemical and pharmaceutical reasons, be turned into drugs. But 15 years later and a bit more, we finally invented something which looks very much like a drug. It's been licensed by Apollo Therapeutics, and it's going to be in man, we hope, in first-in-human testing very soon. Here is the latest results from that project, cryo-EM, looking at the structure of the drug CRP complex in solution effectively. This is the vitrified solution of the complex of the drug and the CRP, and starting with these EM images and doing computer averaging and enhancement and so on, you end up with this 3D structure, which you can solve to very high resolution. This is the 2 angstrom, 2.5 angstroms or so resolution of the drug CRP complex. 2 pentamers of CRP cross-linked by 5 drug molecules. Here's the drug sitting there. It's been patented, so this isn't secret anymore. And here it is. The resolution is high enough to see the individual water molecules. That's what these little red blobs are. Fantastic stuff at the cutting edge of science and medicine. Now, Vittorio has made 2 absolutely seminal contributions to amyloidosis. Many other very important things, but 2, in my opinion, really seminal. One is the importance of lysozyme and the mutations that were destabilizing in lysozyme, which I've already told you about. The other discoveries are related to mechanisms of amyloid fibrillogenesis, mechanical and enzymatomechanical forces. So within months of arriving in our center, this wonderful paper describing a single family with a new, in fact, the first mutation ever found in beta-2 microglobulin, which is very amyloidogenic. And we predicted in writing this abstract, this knowledge should allow further elucidation of fibrillogenesis and protein misfolding. And sure enough, it did, because Vittorio and his team went on to show that our results elucidate the molecular pathophysiology of amyloid deposition. And it's mostly to do with shear flow, hydrophobic surfaces, in other words, mechanical forces. This was a brilliant original insight. Now, meanwhile, after his time in my lab at Hammersmith in 1995 or so, when he spent a year with us, Vittorio in 2004 wanted to come and do a sabbatical in my lab and said, what should I work on? What do you want me to work on? I said, well, I've just invented some new molecules which are intended to be drugs to target TTR and stop TTR amyloidosis. I want you to work on that. So he came and worked and made absolutely crucial contributions. These molecules, which I had serendipitously invented, they enter into TTR. This is the TTR molecule. This is the drug. They enter into it. They don't just stop in the binding pocket where the thyroid hormone is held. They go all the way down the molecule and occupy both binding pockets and the channel in between. And these molecules are super stabilizers of transthyretin. They completely stop it under any circumstances, turning into amyloid. It was a really crucial observation. And Vittorio went further. He showed that the actual way in which TTR makes amyloid in vivo, in both in vitro and in vivo, requires mechanoenzymatic forces. It requires mechanical forces to predispose TTR to being cleaved by tryptic proteolysis and yield a fragment which catalyzes both the intact and the cleaved fragments to make amyloid. And what's more, he identified, together with Alessandro Corazza, another collaborator, and his whole team, by brilliant inductive logic, they identified plasmin, the key enzyme of the fibrinolytic pathway, as responsible for amyloidogenic cleavage of TTR, both in vitro and in vivo. Just unbelievably brilliant stuff. Here's the series of papers, each of which have got their last sentences predicting the next step. I won't bother talking about those now. Here is work on the drugs, our potential drug versus the existing so-called monovalent TTR stabilizers, which like tafamidis and AG10, which do very little. In fact, AG10 does absolutely nothing. And so, you know, we totally confidently predicted these failures, and we've been right. And here, the latest brilliant iteration, this is Alessandro Corazza's NMR work, looking at residue by residue, the impact of ligand binding, drug binding on the TTR structure and stability, confirming the superiority, the potency and superiority of our so-called bivalent superstabilizing ligands. So our lab achievements over this period, 23 years, 22 and a half years, I guess it is now. How has it been achieved and what have the outcomes been? Well, I have won a lot of peer-reviewed competitive research funding, more than £25 million in total during this period, from the MRC, from the NIHR, from the Wolfson Foundation, from the Wellcome Trust, British Heart Foundation, and other bodies. We've also had very substantial commercial funding, the huge program with GSK to show the antibody immunotherapy worked. And following that, we had another multi-million program working on the superstabilizers, which they also abandoned prematurely. Typical big pharma, I'm afraid to say. They have other horizons than the ones that we have, where ours are much narrower. We just want to make a drug at any cost. They need to look at the cost of the drug and the commercial implications and so on. So those stop. But now we've got Apollo supporting the CAAPP inhibitor program. We've got the UCL Tech Fund that's funded $1.5 million for the superstabilizers, together with the guidance and support of Cengiz Tarhan, who founded Free Medic and then UCLB. I founded a UCL spin-out, Pentraxin Therapeutics Limited, which was commercially successful, ended up owning more than 30 patents on my inventions. And other outputs have been 87 high-impact original papers, 28 chapters and reviews, and major new potential drug discoveries and developments. Some, and I hope several of which, will eventually come to fruition. We've been recognized, and I in particular have been recognized for this, and I just highlight a couple of things. The Harveyan oration, it happens once a year since 1656. I gave the 351st Harveyan oration at the Royal College of Physicians. Here's William Harvey, discoverer of the circulation of the blood, the founder of all modern science, the first person in the world to hypothesize about what was happening in a world process, to do experiments to test the hypothesis and to prove it. The circulation of the blood, William Harvey, foundation of modern science. So this is a really great event. It's the primary lecture of the Royal College of Physicians, and that's me and Harvey and Ian Gilmore. I've been honored to be knighted by the Queen for services to biomedicine, and of particular significance to me, in 2014, my alma mater, Trinity, brought me back again as an honorary fellow for the rest of my life, which is a considerable accolade. So what's happening now? Enough history, what is happening now? The BRC core funding, which has sustained the Wolfson Drug Discovery Unit since 2012, ends on the 31st of March and the unit closes. I retire from my paid UCL post, but I will remain Emeritus Professor of Medicine to direct and supervise the work needed in UCL for my current UCL-related programs, which, as I've told you, are the DESPIAD trial of miridisap in Alzheimer's disease, that's funded by About £5 million of NIHR money has been funded by that. That's still ongoing. The CRP inhibitor drug development, many millions funding from Apollo. The TTR superstabilizer drug development program, licensing discussions are in progress on that. What else is happening? Well, very sadly, tragically for the UCL and the Royal Free, we've lost Vittorio. He's retired and is now a scientific director of the San Matteo Hospital in Pavia, one of the largest hospitals in Italy, a very high profile job on which we wish him the very best, but he is remaining a UCL emeritus professor with his office in the center, and he will continue to direct and supervise Diana and Guglielmo. Graham's going to retire on the 31st of March. He will have an honorary appointment thereafter, so he'll be able to work at UCL as and when he wishes. Patrizio will resign on the 31st of March and return to Pavia. Stefan will remain in the center on a new UCL contract for a while from the 1st of April. And I will continue from my emeritus post to direct some work by Graham, Guglielmo and Diana to secure and protect UCL's major investments in our inventions, which UCL has patented and paid for and so on. So let me finish with an acknowledgement slide. Of course, I want to acknowledge the MRC and the NHS, the NIHR, the Wolfson, and all our other funders. We couldn't have done this without their generous funding. The patients, we do all this work for the patients, and without them, there wouldn't have been any such work. I want to acknowledge my laboratory and clinical students, colleagues and collaborators. They're all cited as co-authors of the publication, so anybody can see who they are and who they have been. And I want to name specifically a small group of people for reasons of chronology and particular affection. Jeff Herbert made my first sheep antiserum in 1972. That's a long time ago. And later he came to work, he worked at Babraham, then in Cambridge. He came to work for me first at the Hammersmith, and when we moved to the Royal Free, he came there before he eventually retired. Glenys Tennant came from New Zealand to work for me in 1979, and she was with me for 40 years. She was my strong right hand. Words cannot describe the contribution she's made to me, my life, my work, everything that I've done. Absolutely indescribably wonderful. Winston Hutchinson was a strong rock. Turn his hand to anything you asked him to do. Reliable, efficient, competent. Patrizia Mangione has been a strong right hand to Vittorio. She's been amazingly helpful to me. She has a telepathic knowledge of where Vittorio is at any time during the 24 hours, 365 days a year. I don't know how she does it across continents, across the atmosphere, but she's been invaluable in that as well as many other respects. Graham has been a close colleague, friend, collaborator for decades. We couldn't have done huge chunks of what we've done without him. He's made an invaluable contribution. Likewise, Stefan, who took over the animal work in 2006 or so, and has been responsible for carrying it forward from then until now. And finally, Islam, our IT go-to guy. Everything depends on him. He is invaluable to me, as I'm sure he is to all of you. He is the most outstandingly helpful person one could possibly imagine. And finally, I want to mention and highlight Vittorio Bellotti. He is a great scientist. He has been a wonderful colleague, collaborator for many years, and he is a true and loyal personal friend of the highest order, and I give him the greatest respect. That's it from now, chaps. I'm happy to answer any questions that anybody may have, either now or in the future, in relation to anything I've said, anything that anybody wants to know more about. You can email me, you can phone me, and either now or after you've looked at the recording. So thanks very much.